Hello everyone. In this class, we are going to discuss about the nested PCR. Nested PCR was developed to increase both the sensitivity and the specificity of the PCR. And here in this technique, we are going to use as the two pairs of oligonucleotide primers and two rounds of PCR. Regularly, as we discussed in the earlier classes, in the PCR or in the polymerase chain reaction, usually one pair of oligonucleotides are going to be used, and here we are going to perform the PCR. But here in this nested PCR, we are going to use the two sets of oligonucleotide primers, and here these two sets of oligonucleotide primers are going to be used in the two different rounds of the PCR. And here typically one primer pair is used in the first round of amplification of the PCR of 15 to 30 cycles. The products of the first round of amplification are then subject to a second round of amplification using the second set of primers. So that means, so here in the first round of the PCR, the first amplified product will be formed. That the amplified product which is produced in the first round of the PCR is transferred and exposed to the second round of amplification. And here in the second round of amplification, the second set of the primers are going to bind to the amplified product which is formed in the first round and going to produce the amplified product in the second round also. So that here the second set of primers anneal to the sequence internal to the sequence amplified by the first primer set. That means, so here in the first round, if the amplified product is formed, then only it can be used in the second round of the amplification. And here in the second round of the amplification, the second set of primers will bind to the region which is present or internal to the amplified product which is formed in the first round. So that here it is going to then only the second set of primers are going to produce the amplicon or the PCR product. Okay, the increased sensitivity arises from the high total cycle number, and the high specificity arises from the annealing of the second primer set to the sequences produced by the first round. So here this technique or this PCR, it is having the higher sensitivity when compared to the regular PCR. It is because it is having the higher total cycle number when compared to the regular PCR. At the same time, it is having the high specificity. It is because in this case we are going to use the two sets of primer pairs, so that here in this case, in the second round of the PCR, the second set of primers are going to bind to the amplified product which is produced in the first round and going to produce the final amplicon or the PCR product. And here, due to which the specificity will be increased in the nested PCR. And here, apart from this one, the major concern for this method is the contamination that occurs during the transfer of the first product to the second tube for the second round of amplification. So that means, so here, as I told you earlier, in this technique, there are two rounds of amplification of the PCR is there, and here during the transfer of the sample from the first round PCR product into the second tube. So here, in that case, there are chances of the contamination is there. However, it can be avoided physically by separating the first and the second round amplification mixtures with a layer of wax or the oil. By doing so, we can avoid the contamination over here. And here, in this case, we are going to discuss about the nested PCR as we discussed earlier. So that here you can see here, in this case, it is nothing but the DNA over here, and here to this DNA, the red color arrow indicates that the first set of primers, which is represented as the outer primer F and the outer primer R. These two arrows are indicate that the first set of primers, and here they are specifically command bind over here and going to produce the amplicon, which is indicated in the red line in the first round of the PCR. And here, in any case, if these the first round of the primers, if it is going to bind specifically to the DNA, means it may also result into the non-specific amplification also. But here at the same time, we can see the specific amplified product which is formed in the first round of the PCR. So here, after the production of the first the specific amplified product in the first round of the amplification or the PCR, now it is going to be exposed to the analyze to the second round of the PCR by using the second set of primers which are indicated in the orange color. So here they are named as the inner primer F and the inner primer R. So that here these primers are going to bind to the region which are internal to the amplified product which is formed in the first round of the PCR. So that means these second set of primers, they can bind to the amplified product if these amplified product only if it is formed in the first round of the PCR. In the first round of a PCR, if this amplified product is not found means, so here then these internal primers will not bind to the amplified product in the second round. So that here it is going to assures the specificity over here. In the second round of the PCR, these the second set of primers, inner primer F and the inner primer R, bind to the amplified product which is formed in the first round of the PCR internally with inside the amplified region, and ultimately going to produces the amplified product in the second round. And here by the use of the two sets of the primer in the nested PCR, we are specifically amplifying the specific target region in the sample, and by doing so we are avoiding the amplification of the non-specific amplification in the second round of the PCR. In the second round of the PCR, the specificity will be much more increased. So that here this is the principle of the nested PCR. Now we are going to discuss about the applications of the nested PCR, and here to improve the sensitivity of the assay, nested PCR has been used in the many PCR assays. It is particularly useful for suboptimal nucleic acid samples such as those extracted from formalin fixed paraffin embedded tissues. So if any samples is fixed to the all these things means, so here in that case they will be having the very lesser concentration of the nucleic acid. So here in that case we need to have we need to do the PCR which is having the higher specificity and also the sensitivity. At that time we can choose for the nested PCR. And here apart from that one, the nested PCRs have proven valuable for the detection of microorganisms when they are present in very low quantities. For example, in the detection of the rickettsia, Bartonella, and similar organisms in the blood and the tissue sample. So here these pathogens which are present in the samples in the at the low quantity. So here at that time we need to choose the PCR which is having the high sensitivity and also the specificity. At that time we can choose for the nested PCR. At the same time it is also going to be helpful in the detection of the herpes virus, enterovirus in the CSF, and also in the detection of the mycobacterium tuberculosis in the sputum sample. Thank you.